Hi everyone, this is all about the breakdown of molecules in the digestive system. It's all about making big things into smaller things so they can get into the bloodstream. So there's quite a bit going on here. Most of it is, like I said, just about absorption. And we also need to know the main organs of the digestive system. Page 38 to 39 helps you. I've got a few pictures on here which will also help sort of to embed that as well. So it starts off with carbohydrates. Carbohydrates are our fuel. That's what we run on. We need to know the formula for glucose, C6H12O6. It's relatively easy to remember once you get your numbers right. So 6 on the C, 6 on the O, and together they make 12 hydrogens. So it's quite a good way of just remembering it. It's a quick way of writing glucose as well. You can do that in exams unless it asks for a word. The main problem with the digestive system is we can only get small sugars like glucose and fructose there into the bloodstream. Larger sugars have to be broken down into those smaller ones before they can be absorbed. One of the most common long chain molecules is starch, which we can absorb directly after it's been broken down, because all it is lots of glucose molecules joined end to end. Cellulose, we know it as fiber, we can't digest, it passes through us. Only certain animals can do that, such as rabbits, horses, etc. Even they have difficulty doing so. They have to re-chew the food, chewing the cud, to allow them to break it down enough so they can absorb the glucose. Lipids are slightly more complex because they're actually got four parts to them: three fatty acids and the glycerol shoulders there. This is why they're called triglycerides, because it's got three in it. They are essential for many things. They've got a very high energy store and they're also insoluble, so they don't affect osmosis. And we need to break them down so that all the components can be absorbed. What happens is enzymes work on the fatty acid tails there and break them off from the glycerol. So we end up with glycerol and the three fatty acids being absorbed into the bloodstream. Together, this is too big to be absorbed. But if we break it apart, it can move in through the very tiny little gaps in the blood vessels. Next one is proteins. Proteins I like to think of as like Lego. There's loads of different shapes of them, and you can fit them together in different ways to make different things. Proteins are incredibly versatile and are very abundant in nature. They're one of the main molecules we find in the natural world. They have lots and lots of functions, such as building cells and tissue, so growth and repair. They make hormones. Most of our hormones are proteins. Our antibodies are proteins. And the biological catalysts that help to speed up rates of reaction, enzymes, are all proteins. So proteins are incredibly versatile. They're made up of about 20 different amino acids. It depends where you look. There's different types because some have subunits and stuff like that, but 20 is the accepted generic number. And what happens is you combine them together in a particular order, a lot like Lego bricks. You put them together in a particular order, following a pattern, and it will make a house or a car or whatever. The same with this. You put the amino acids together in different sequences, and they will make different proteins. They are incredibly complex molecules, highly folded in this unique 3D structure, and the bonds are very susceptible to temperature and pH change. So at high temperatures and high pH, what happens is those bonds break and the protein becomes denatured. That's the correct term for it. You cannot kill a protein because it's a molecule, it's not alive, but it can be denatured. There are many key bits of the digestive system, but in a nutshell, we need to know the following in a little bit of detail there. So all these structures have to be sort of at least partially known. Often this is a labeling activity, so they'll give you a picture and they say what is X, what is Y, but sometimes they ask you to explain a little bit more about the function. So it's worth sort of learning it from mouth to anus. To help you, I've provided you with a picture here. You can pause the picture just to get the key points down. As a word of advice, the American spelling here creeps in quite a bit. We actually spell it starting with an O, but it's just something that we should be careful about because correct English is important in exams, so we don't want to slip into Americanism. And the last bit is a nice little fact to finish off on. It's those amino acids. Like I said, there's about 20 of them. And as you can see, the basic structure of each of them is the same, but the bit circled in green here is different on each one. That's actually called the R group. It's the group that changes. The amino bit and the acid bit stay the same on each of them, but it's this green bit that changes. So they're all unique, exactly the same way as I said, like Lego, you've got different size blocks and stuff like that. Anyhow, hope that helped, and I'll be making a few more of these. Stay safe. Bye.